In this video, we will cover other SARS-CoV-2 variant that are significant or that have a significant impact on the world. Uh, the next one or the first one for this video is Delta variant. And this Delta variant was first found in India in April 2021. However, in USA, the first case was found in March 2021. And this has around 10 mutations in the spike protein that are listed here, and this is one of the uh, strongest variant at that point of time that have highest infectivity and less neutralizing, and these are less neutralizing by the antibodies. So this Delta variant was the major cause of concern at that point of time. However, when the Omicron arrives, it is the infectivity on the infectious is far worse than the Delta. We can see that it is 13-fold increase in case of infectivity. Here it is typing mistake. It should not be 13 times. Viral infectivity, you can consider it from here. Viral infectivity is 13-fold increase and 2.8 times more infectious than the Delta variant. And this has first spread in the South Africa, and after that it spread all over the world. So Omicron variant so far has been considered as the major infectious SARS-CoV-2 variant, as it has 30 changes in spike protein, and apart from that it also has changes in envelope nucleocapsid protein and terminal domain, receptor binding domain, and non-structural proteins. And after that, let us understand whatever the things that we have discussed. We need to understand why we have included only this variant, not the other variants. The main reason is that variant of concern. WHO define any variant as a variant of concern, and when its infectivity is more, and the neutralization capacity of the antibodies is less for that variant. Otherwise, that variant is considered as variant of interest. And here is the structure of the spike protein of SARS-CoV-2, and this structure can be accessed through RCSB website, where we need a PDB ID, and the PDB ID for this spike protein is 6VSB. And so this is the three-dimensional structure, you can say that, and this is the linear structure where it describes every domain, such as it starts with the signal sequence. This is the N-terminal site and this is the C-terminal site. After that it is N-terminal domain and receptor binding domain. Then it is subdomain 1, subdomain 2, and here it is the cleavage of S1 and S2, and this part is known as S1 subunit. The rest of the part is known as S2 subunit. And all these details, that is fusion peptide and C-terminal domain, transmembrane domain, uh, can be found in details from the reference paper that is presented here, and this link has also been available in the description. And here is the this is the combined structure of spike protein, and here it has been separated out the structure of spike protein. That is green color domain is the N-terminal domain, and yellow color is the receptor binding domain, and pink is the subdomain 1 and subdomain 2, and purple we can say is the uh, it's a part of S2 subunit or S2 uh, subunit of spike protein, and this is we can say this is represented as the receptor binding domain. C part is the receptor binding domain in detail, and this can also be accessed with the 6VSV in the when we need to see the upside consideration, and when we need to see the down configuration, we can use the PDB ID as 6VXX. So in this case we can access the three-dimensional structure from RCSB website, and if someone wants to access this structure on their own computer, then they can use the software such as PyMol, where we only need this ID to fetch the three-dimensional structure of the protein. Of course, I guess we also need the internet connection, but I'm not sure about that. So yeah, for the last two videos are only about the informations, and that does not involve too many structures. That might be the reason that people or people might not be that much interested to understand, or that much interested, or these videos are not that much capable enough to understand people, or to make people understand about the different variants in an interesting manner. However, if I found some other method to describe these things in a more pictorial form, then I will publish another videos with that new methodologies that might help you to understand these variants in an interesting manner. So that's all for this video. Thank you for your time.